And you can see this if you actually look at SEM pictures of Futsi-Taratsu mutants. So here is an embryo that's wild type. You can see all the different segments that have been formed. And if you look in a embryo that has mutated Fushi-Taratsu gene, it has fewer segments. And these lines kind of delineate where the normal segments would be. So you can kind of see that this mutant has a pretty large section, and in the wild type, this would be divided into 2 segments. Okay, so missing one of the parallel genes really messes up the pattern. Yes.
So are those segments all even skipped?
So all of those would express even skip. Okay. Okay, and so this is important because the parallel genes control the expression of the segmentation genes. And the segmentation genes are the ones that help to start defining these segments of the embryo to specific parts. Okay, so last time we started talking about how these segments become defined by boundaries. And we talked about parasegments versus segments. And this is the graph I showed you where you're looking at expression of parallel genes, so Eve or Fushi-Taratsu, in the context of a single parasegment. So for example, here, Eve expression is high towards the anterior, but it's missing in the most posterior part of the parasegment. And so this pattern, where you have a parallel gene missing in the posterior sections of the parasegment, results in the expression of wingless in a cell or a group of cells at the posterior end of the segment. Between parasegments, there's another gene called engrailed that's expressed in the most anterior part of the parasegment. So the wingless expressing cell and the engrailed expressing cell are neighbors. So between parasegments, you have these patterns of alternating wingless engrailed, wingless engrailed, wingless engrailed. So this is the beginnings of boundary formation. Okay, so what are these genes? Okay, wingless is a ligand. It's we'll be talking about Wnt when we talk about vertebrates. So wingless is basically the equivalent to Drosophila Wnt. And this is the diagram from your text that shows you the signaling pathway. So Wnt, or wingless, will bind to its receptor. The receptor is called frizzle. And when it binds, it starts a cascade of events intracellularly, and you get the expression of a protein called beta-catenin that translocates to the nucleus and activates the expression of Wnt target genes. And so a target gene is basically just a gene that is activated by a specific extracellular signal. So here a Wnt target would be, for example, hedgehog and engrailed. Okay, hedgehog is another ligand that binds to a receptor called patched, which is shown here in brown. And for now, don't worry too much about all of this on the side here. But essentially, when hedgehog binds to its receptor patched, it results in a cascade of events that allows this gene, CI, to be translocated also into the nucleus and activate hedgehog target genes. And in this case, an example of hedgehog target genes are wingless and engrailed. Okay. So wingless activates hedgehog and engrailed, and hedgehog also activates wingless and engrailed. And so if we look at these two signaling pathways in the location between the parasegments, you start to see positive feedback loop. So this is the cell that expresses wingless. The protein is excreted into the extracellular space. It binds to a cell that expresses its receptor frizzled. You have a cascade of events which results in the transcription of its target gene engrailed in hedgehog. A hedgehog, in turn, is secreted, and it binds to its receptor, patched, which results in the translocation of this protein, CI, and turns on transcription of wingless. So you get this positive feedback loop, which helps to basically cement that this cell expresses wingless, and this cell expresses hedgehog and engrailed, and that this is the boundary between the two parasegments. Okay? Okay, so if we kind of zoom out now and look at the parasegment or the segment again, we have our gene that expresses wingless, and we have our cell here that expresses engrailed, and this neighboring cell also expresses engrailed. Here you see the positive feedback loop that I showed you earlier. Okay, so if we look at the parasegment, we see that some of the cells in the anterior express the receptor for hedgehog patched, but some don't. And we also see kind of these arrows of diminishing width, and this is supposed to represent diffusion of the wingless protein from its source. Okay, so now again we're kind of come back to the idea of concentration gradients and morphogens in the parasegment.
Would there be some production of the wingless protein in the neighboring cells that have the patch receptor?
So yes, there should be. So if hedgehog induces expression of wingless, these should also express some.
It's just a gradient.
But yes, so there's a gradient of hedgehog expression too. So this is showing that the hedgehog is expressed at about equal levels, but if you think about it, the concentration is getting more diminished as you travel further from the source. Okay, so it's thought that this diffusion of the wingless protein and the hedgehog protein is important for defining what these individual cells in a segment will be. And it's not really worked out well, but there are some data that support that hypothesis. So, and that's shown here. So this is a wild-type embryo, and here we're looking at one segment, the A3 segment. And so if we look at the A3 segment under SEM, what we can see is that there are basically 4 regions of this based on the hairs that are expressed there. So the primary region has a few hairs, the second one doesn't have any, the third has a few more that seem to be pointing kind of towards each other, and the fourth region is more wide and the hairs are pointing towards, basically polarized towards the back. And then you have the primary region of the next segment. So it's thought that the different gradients of hedgehog and wingless that are expressed in these specific regions control what kind of hair pattern they will have. And there has been some experiments done where if you change the concentration of hedgehog and wingless in the embryo, that these patterns change. But it's still kind of confusing to think about how all of this actually works together. So this is still a work in progress to understand how the hedgehog and wingless gradients within a segment resolve